So my name is Ken Welch, and I'm speaking on behalf of the postdoc that I had do this work, who is tending to her, I think, 4-month-old baby now. And that person is Julia Rossi, a fantastic researcher and holder of NSERC and L'Oreal UNESCO Fellowship. And first, I'd just like to acknowledge that we did carry out this work in Belize on the traditional lands of indigenous people who have historically been displaced from their territories. And we honor with gratitude the land itself and the people who have stewarded it throughout the generations. And that applies not only to Belize, where we did this field work, but to where we work in Toronto, where we analyzed this data, et cetera. So my lab has long been interested in sort of the interplay of dietary specialization and metabolic physiology. And when we think about running mammals, there is a long understood, highly conserved pattern of the fuels that we burn when we exercise. And that is that at lower exercise intensities, we rely to some great extent on lipid oxidation. But as our exercise intensity increases in humans, mice, rats, horses, dogs, et cetera, we see a progressive shift towards carbohydrate oxidation, and that's primarily in the form of internal stores of carbohydrates like muscle glycogen. So again, this is a broadly conserved phenomenon among vertebrates, including our puppy Charlie here. And that relates to the fact that our diets among most mammals are usually complex and filled with glucose and carbohydrates, which makes glycogen, which is good for fuel. But in other species, particularly pollinators, their diets are quite specialized to be very rich in just simple sugars. Obviously, there's some need for amino acids and other micronutrients, but calorically speaking, for specialized pollinators, a lot of the calories coming in is in the form of simple sugars. And so we see convergence in behavior and aspects of phenotypes amongst both invertebrate and vertebrate pollinators, including the mammals, including nectar bats, which are pollinators. And so again here, we're seeing this specialization on sugar leading to a specialization in fuel use corresponding to that. These animals show a remarkable ability to utilize dietary sugar, their nectar sugar, to fuel their intense exercise. And we can visualize that phenomenon here. This is a breath stable isotope tracking approach. I'll get into that just a bit more, but essentially the y-axis here is showing you the proportion of exhaled CO2 that is derived from the fuel of interest, a tracer fuel that we've given them. So we give them a bit of stable isotope enriched sugar to drink, and then when they come and feed from the feeder and rebreathe into the mask that we give them, we can estimate this contribution of that dietary sugar to their exercise. And you can see that, that in the hummingbirds and bats, we see a very rapid increase and a reliance, almost completely in some cases, 100% on that sugar. And contrast that to what you see in humans. The Gatorade Institute would like you to believe that drinking Gatorade will dramatically increase your performance. But that's a bit of an overstatement because you just don't have the machinery to import those dietary sugars into your body and begin using them that quickly. So humans are down here and they're embarrassed by these more aerobic animals. But there's many ways that diets can be specialized. Nectarivores have a very sugar-rich diet, but in contrast, blood-feeding invertebrates, tsetse flies, mosquitoes, etc., their diet is very distinct, at least female mosquitoes. We can distinguish between male and female mosquitoes here. It is a blood diet. It is protein-rich, carbohydrate-poor, but they have high metabolic rates. They are hovering or flying animals, and it turns out that research has shown that in these animals, they are incredibly good at oxidizing not carbohydrates or lipids, but amino acids to support that intense exercise, such that when a tsetse fly runs out of proline, it has to land and feed again before it's going to be able to fly very much. So bats make a really excellent system in which to explore this interplay between dietary specialization and physiology because they have evolved in rapid succession so many diverse diets. The original bats were thought to be insectivores, of course, like the big brown bat you might find here in the Montreal area. We have fruit bats, which obviously specialize on a sugar-rich diet, our nectar bats, various flavors of those, but we also have examples of the few vertebrates that have evolved to become obligate sanguinivores, vampire bats. They eat blood and nothing else, period. And that got me thinking, Well, humans are really bad at using amino acids to support aerobic exercise. We're set up to use carbs or lipids. No more than 5 to 10% of our metabolism can be supported by amino acid oxidation. But in the tsetse fly, it's around 100%. So I wondered, do we have examples of convergence here in metabolic physiology because of this similar specialization of diet? To assess this, as I mentioned before, we use stable isotope breath tracking. We can do respirometry and look at the rate of oxygen consumption versus CO2 production. That tells us something about the nature of the fuel being used. We can somewhat distinguish between carbs and lipids, but it doesn't allow us to fully distinguish because when we throw amino acids into the mix, it's difficult to partition that out. So we need a tracer. And in this case, we're using leucine and glycine in our particular study. And regardless of the fuel, carbohydrate, lipid, or amino acid, if it is fully oxidized to CO2, the carbon that was in that fuel molecule is the carbon that's going to be in the CO2 you breathe out. So if we give a labeled, carbon-13 labeled leucine or glycine, then we can potentially measure the appearance of those heavier carbon-13 O2 molecules in the breath. That's easy enough to do when we're talking about a human where we can ask them to wear a mask and get on the treadmill, or to even a hummingbird or nectar bat where we can invite them to come up to a feeder and voluntarily insert their head into a mask to breathe for us. But these are animals that need to be able to hover to do that. I don't have access, at least not in Belize, to a wind tunnel in which to study animals that cannot hover. We need more room there. But vampire bats are also fairly derived among bats in having very capable locomotion on land as well. That is that they have independently evolved their own running gait, which is similar to what you'd see in gorillas, or if you have ever had an injury and had crutches and were trying to catch the bus. So that means that I can get to my shameless plug of doing this great work at the Lamanai Outpost Lodge. This has been featured in the last few weeks on NPR, the Batathon. I encourage you guys to look that up. And I want to give a special shout out to Sable Systems International, which allowed me to borrow their prototype rat metabolic treadmill, which was big enough to accommodate our vampire bats. Vampire bats are incredibly intelligent, more so than rats, I would say, and they quickly figured out that there were lots of little holes where they could stick those thumbs in and cheat. Just hold on. But with some duct tape, we of course fixed that, and they could, ooh, I'm sorry, that's very jumpy there, but you can see that this guy's happily hopping along or running along at 30 meters per minute, and it will do this for hundreds of meters. They are incredibly good on the ground, as they need to be to approach their prey and come up close and sneakily nip and start lapping that blood. So what did we do? Like I said, we gave these bats either a control diet of cow's blood that was not enriched with our carbon-13 isotopic amino acids, or ones that were enriched with, or spiked rather, with the tracer amino acids of either leucine or glycine. One is an essential and the other is a non-essential amino acid. Here you can see that the bats ate similar amounts of each of those. The exercise protocol involved feeding them, and then after a few minutes, putting them into the metabolic treadmill, before which 4 minutes we got a baseline without the bat in there, so we had our background levels of oxygen and CO2. We put the bat in there and we ran them progressively at 10 meters, or walked in this case at 10 meters, that should say minute, excuse me, 10 meters per minute, 20 meters per minute, or 30 meters per minute, and then we repeated that 3 times. So these guys are really quite aerobic and good at this. And what you see on the right-hand side there is measures of oxygen consumption and CO2 production. And as we would hope to see, if they are running faster, their metabolic rate is higher. And you can see that the magnitude of change there is relatively similar between the two and not different between the treatments there, indicating that the overall fuels being used were similar. So this respiratory exchange ratio, this ratio between CO2 production and oxygen consumption, was very consistent during the trial at between 0.8 and 0.9, which is consistent with the RER you'd expect from the oxidation of amino acids. But as I said, carbohydrates and lipids also lead to characteristic RER signatures, which confound our interpretation here. So this is consistent with amino acid oxidation, but not sufficient to prove it. The raw isotope tracer data, the enrichment of that carbon-13 signature in the CO2, follows this pattern. It is highest during the initial measurements, of course, and then it progressively declines over time. And a couple things to take away from here. This is a very rapid appearance of those amino acids in the breath being oxidized. This is within minutes, almost instantly, it seemed. And then it declined over time. These guys had only the one meal at the start of the experiment. You can see that the control values never changed because the background isotopic signature was there, but it's much enriched in our treatments. If we look at the proportion, that FEXO value, the proportion of metabolism that seems to be supported by the oxidation of our labeled amino acids, we find that they are actually quite similar, and we are seeing apparent oxidation rates, or apparent contribution of those ingested amino acids accounting for as much as 60 to 80% of their overall metabolism during those first few moments of walking and running. That's huge. That's way more than we would estimate or measure in humans. It's much more like these invertebrate blood feeders. And it's at least 50% during the most intense exercise here of 30 meters per minute that we subjected them to. More importantly, it's suggesting that the use of essential and non-essential amino acids as fuels, that the bats don't care. They're happy to use either. We would be surprised by that because essential amino acids are usually limiting for us. They come from our diet or from our microbiome. It's not limiting to a bat when all you're doing is drinking blood all the time. So in conclusion, we find that the metabolic rate of running vampire bats can be reliably measured, which gives us a neat tool to begin to investigate their energetics more extensively, and more analyses are to come there. And that, as we suspected, vampires rapidly and extensively oxidize amino acids to fuel running and likely flight, though we couldn't test that here. And they are more like a tsetse fly or a female Aedes aegypti mosquito than they are most self-respecting mammals. And further, that they don't seem to discriminate between these two classes of amino acids, which is also quite surprising. Just a caveat, I would say that we are probably overestimating the contribution of the amino acids to overall fuel use, because the diet that we gave them was cow's blood. It was not simply a mixture of individual amino acids, which we spiked that with. So it's entirely probable that the oxidation of the more complex proteins in that blood meal took a greater amount of time. Nonetheless, I think we have strong evidence here that these guys are rapidly making use of these amino acids to fuel a substantial portion of their metabolism. And we want to follow up with this and to try and figure out the difference between oxidation rates of individual amino acids versus the more complex substrates that are in the blood. With that, I would like to thank our collaborators, hosts, and you for your attention, and I'm happy to answer any questions. Sorry, this is the most viral tweet I've ever been associated with. Julia posted this, and it's over 100 and something thousand views. So, any questions? Oh, yes.
Hi, hello. I have a question. Maybe this is a little bit out of the investigation you did here, but is there any difference between the blood of a cow versus like wild animals that they may be consuming?
There are certainly differences. Are they germane to the question we're asking? Not as clear. I would assume that there's going to be similar amounts of free amino acids and complex proteins making up the blood of capybaras compared to cows. There are vampire bats that specialize on avian prey versus mammalian prey, and there you might see some differences as well. But the question we're asking at this point, I think, is a little above that granularity. It would be interesting to compare amongst, there are 3 species of vampire bats, and it would be interesting to compare amongst them and see if there are differences in their handling.
Okay, thank you.
All right. Thank you all very much.
Hi, all. My name is Nara, and I'm a postdoc in the mechanical lab at the University of Memphis. And today I'm going to talk about the predicated protein structure and the phylogeny, confirm the convergent evolution of GH28 in the families Cerambicidae and Bupricidae. So before I go talk, I want to give like a quick introduction and a summary of the previous study. So regarding the feeding behavior, beetle is known as generalist to the specialist, but most of the beetles are phytophagous beetles. And these 4 groups contains many number of the phytophagous beetles. And their main food source is plant. And the plant cell wall is mainly composed of the plant cell wall. And this structure is heterologous and rigid. And So this contains the abundant polysaccharides such as pectin, lignin, cellulose, and hemicellulose. And actually, this component is really difficult to break down. So when beetles eat the plant, they need a special digestive enzyme to break down these polysaccharides. So we call this plant cell wall degrading enzymes. And most insects rely on symbiont to obtain this enzyme, but some beetles family produce it on their own. And this Buprestidia and the Phytophaga are the family and also group. And among them, the Phytophaga is well studied about the evolution of the plant cell regulating enzymes. So Phytophaga acquired plant cell regulating enzymes via horizontal gene transfer event from different microbial donor. And in the ancestor Phytophaga, they acquired the pink set of the plant cell regulating enzymes from different different microbial donor via different HD event, and they pass it on to their descendant. And subsequently, during the differentiation into modern lineages and also phylogenetic divergence, the multiple HD event occurred that transmitted variant plant cell regulating enzyme horizontally. And among the Phytophaga's plant cell regulating enzyme, today I wanted to focus on GH28. So ancestor Phytophaga, they acquired this GH28 from the Ascomycota as a putative donor, and they passed it on to their descendant. And also this gene encoding the enzyme gained intron, and most of the present Phytophaga beetles possess the ancestor type GH28 with this intron. But one cell family of the Cerambicidae, Laminae, they lost it but acquired it again from different Ascomycota as a putative donor again, but it does not contain any intron. So I think maybe you just have some wonder what the GH28 is. So GH28 is glycoside hydrolase family 28, and is a really well-known endopolygalactonaceous in fungi. And this endo-PG is a breakdown pectin. Pectin is composed of the really long polygalactonic acid, and this endo-PG break down the polygalactonic acid randomly to produce oligogalactonic acid. So Ascomycota GH28 is also well-known endo-PG, but in nature also we have exopolygalactonaceous, exo-PG. It breaks down the glycosidic bond from the non-resisting end to produce mono and digalactonic acid. So why I talk about the exo-PG? Because as I mentioned, the Ascomycota is a really well-known endo-PG, but previous study confirmed that Laminae GH28 shows endo-PG activity as well as the exo-PG. So this is a result of the neofunctionalization via evolution, and also this gene has evolved to required for the special diet necessary on this beetle. And also in the previous study, we confirmed that Laminae is not only the cell family possessing this new type GH28, Buprestidae also has it. So these two, the Buprestidae and the Phytophaga, diverged from 250 million years ago. So how it happened? But it's not that surprising because the Cerambicidae and the Buprestidae, they have a similar developmental stage in their life. Both are the wood-boring larvae and also Phytophagous beetles. So this led me to having some question. First, did this enzyme originate from the same HGT event, or did this GH28 undergo convergent evolution to adapt to similar environment? So to figure out, I worked with this following method. First, I annotated the gene and analyzed the gene structure using NCBI Buprestidae chromosomal level genome dataset, and also I compared the microsynteny between these two families and within each families. And last, I also employed AlphaFold 2 to predict the protein structure and link them to identify enzymatic function and help me to predict putative function of GH28. So let's talk about the result from this following method. So first, I manually annotated the GH28 from 3 agarose species, including the invasiveness species and the one Tosima for Buprestidae study. And I compared this gene copy variant to the Laminae GH28. So first, I confirmed that Buprestidae possess really wide range of the GH28. And second, this gene copy variant number does not relate to the invasiveness property. And also, this gene copy number does not relate to their host specificity. So I also generated new GH20 gene tree, including this newly annotated GH28 from Buprestidae. So you can find this gene tree on the left top, but I only zoom on the laminae and the Buprestidae clade today to show their evolution. So first, you can also maybe confirm, you can see this tree. First, I recovered the monoclade for laminae and the Buprestidae. But from that, I also learned this laminae and the Buprestidae, they are not sister clade because laminae is more like a close to the fungi rather than Buprestidae. And I do not show up here, but when I compare their genome structure, I could not find any microsyntony between the laminae and the Buprestidae. But still, Buprestidae GH28, they do not possess any intron. So it means we can infer that the Buprestidae and the laminae has experienced individual HD event, but the donor involved are likely the same Ascomycota. So we can move to the next question. Did this enzyme undergo convergent evolution to adapt to similar environment? So I may figure out this question with predicted protein comparison analysis. So for that, I need a really good reference data set, and I found from the protein database one Ascomycota GH28. This is a known endo-PG, and also from crystallization analysis, scientists confirm that their structure is open structure and also displaying endohydrolytic properties. And also this structure has the N-glycosid site. So it's gonna be the perfect reference protein to study insect GH28. So first, I employed AlphaFold 2 predicted laminae GH28 first, and I picked only one species of the laminae, Anoplophora glabipenis, and also different name is Asian longworm beetles, because this species possess 18 large copy number of the GH28, and also previous study confirmed their substrate specificity. So this species possess the exo-PG and endo-PG either. So when I run the AlphaFold 2, as you can see that AlphaFold predicted the protein structure for exo-PG and endo-PG as the same structure. This structure contains open structure at both ends, also endohydrolytic properties, and still N-glycosid sites. So it means from this protein structure, we can only speculate their different function from different sites. So I utilized the RMSD to compare this protein structure to the reference one. So from that, I learned Asian longworm beetles GH28 show more structural difference in proportion to the phylogenetic relationship. And also this structural difference drive the neo-functionalization when I compare to endo-PG1 and endo-PG3 to exo-PG. But still, when I compare the endo-PG2 and exo-PG, still maybe this structural difference are not always associated with it. So because still remained behind question, but today I don't have really time to talk about that, so I wanted to show Buprestidae GH28 first. So just to remind you, so Buprestidae beetle possess a really wide range of the GH28 gene copy number, regardless of the invasiveness. And from the phylogenetic analysis, I learned that GH28 in the Agarelus and the Tosima from different subfamily in Buprestidae are orthologous. And additionally, GH28 and conserved syntony are presented in all Buprestidae beetle genome. So from that, we can speculate that the ancestor Buprestidae species had at least 3 copies of the GH28 due to the gene duplication, and this trait was inherited by descendant of Buprestidae beetle. And also, it means we can infer that maybe functional differentiation already started in the ancestor Buprestidae level. But we still need to investigate their protein structure to just confirm this hypothesis. So I also need to pick only one species for protein structure analysis. So I picked the Agarus malai because first, this is the invasive species in the United States, and also this species possesses 25 copy of the GH28. This is the largest number of the GH28 among 3 Agarus species, and also I can find all 25 copies of the GH28 in all Buprestidae phylogenetic tree. So it can be a good reference work. And when I employed AlphaFold 2 again for this Agarus malai GH28, surprisingly but not surprisingly, still all protein structure looks the same. they have open structure and also endohydrolytic the properties and glycogen site. So still I need to run RMST to compare this protein structure to the reference one. So like Asian long bears GH28, this agaricimolide GH28 exhibit more structural difference in proportion to phylogenetic relationship. But maybe you cannot see here, but when I compare to their RMS score to Asian long beetles, actually these agaricimolide structural difference are relatively more significant than Asian long beetle one. But from that, I still cannot infer their substrate specificity because we still lack of the reference, we still lack of functional calculation data set. So maybe I need to further work for this one. So to sum up, the laminae and the bupresti, they acquired a new type of GH28 via separate horizontal gene transfer event from the same fungal donor, Ascomycota, and the bupresti GH28 result from the gene duplication that occurred in the genome of their common ancestor. And slightly change the protein structure can lead the different enzyme function, and also we can speculate in their functionalization using predicted protein structure from the deep learning machine tool. But still, we need we need to get more like a functional calculation data set to understand the evolution based on this predicted protein structure. So for that reason, and for also other interesting current in the Mechanical lab, we also have an interesting on the evolution of the ancestor GH28, because most of the phytophaga beetles, they possess it, but their rearing environment is totally different from the tropical to Antarctic-like continent. So it means they have evolved in really totally different, like extreme changed niche. So our following question was, how did GH28 evolve in response to various environmental change in phytophaga beetles? So for just for this work, we are currently collaborating with our curriculum lab in the USDA and using insect SF9 cell system to express this enzyme and test their substrate specificity with different plant cell wall poly and oligosaccharides. So I will bring more data set maybe in the next meeting. So thank you for all the lab member and meaningful discussion with the collaborator. So also thank you for listening my talk today. So if you have any questions, so I'm happy to talk about that. Yep.
I was curious, did you look at sequence identity between your different protein structures and if there's any correlation between sequence identity and the RNA differences?
Yes, indeed. So sequence difference is so slightly the change, but when you look at the phylogenetic trees, they are really the close. So hold on. Here. So sequence similarity is similar because it is already reflected in the phylogenetic tree, because it means if they are closer together in the phylogenetic tree, it means their sequence similarity is not that far away. So yeah. So why we thought that this is gonna be like a same HD event, but actually not indeed from that work. Yeah.
Was there a correlation with the sequence differences in the RNA differences at all?
I really don't think so because no, I don't think so. Yeah. Any question? Yep.
Do you have a hypothesis for how the horizontal gene transfer happened?
Yes. Yes, because this Ascomycota, they are just also found in a decayed wood, and these two beetles, they are rearing in a decayed wood when they're larvae, and really for more than 6 months. In case of the Buprestidae, they spent 6 months, and yeah, so this is the reason why they got it. Yeah, thank you for that. Yeah.
Thank you for coming. I'm excited to be here. I'm Danielle, and I'm a postdoc in Suzanne McGaw's lab at University of Minnesota. And this is the first chance I've gotten to present my postdoctoral work. Also, it's my first talk on fish, so I can't pronounce any of the names, but bear with me. I'd just like to start off by thanking everyone who was involved in this project and recognizing that this was a huge collaborative effort and includes also the work of several undergraduates and their theses. I also think it's important to start academic talks out with a recognition of the privilege that helped me do this work, as well as the axes of diversity that posed a barrier to doing this work. And I also want to point out that I come to this conference because it's one of my favorite, but also because it puts out significant efforts to be inclusive and provide resources for people like me to attend conferences. So thank you to those folks. And also thank you, I forgot to thank our funding source, the NSF AP WD. So I'm generally interested in adaptive evolution. And when we think about adaptive evolution, the first thing we usually think about is, with innovation, sequence divergence, these additive mechanisms, regulatory change, gene duplication, things like that. But with the explosion of the genomic era, we've seen kind of an unexpected pattern that gene loss is actually pervasive across Animalia. And gene loss is also associated with traits that we associate with adaptive change, big phenotypes, and in some cases has been directly linked to increases in fitness. Also on shorter timescales, the path to gene loss, relaxation of selection, loss of function mutations, and pseudogenization have similarly been linked with traits that we consider to be adaptive, and certainly big phenotypes that we're interested in understanding. And so it's with this lens that we looked for candidates for gene loss, and this is the work of Erin Gilbertson, who is an undergraduate in Suzanne's lab. She built a bunch of cool custom pipelines and used NCBI and Ensemble to blast genomes, transcriptomes, and raw reads and come up with a really cool set of gene loss candidates. And of those gene loss candidates, there were quite a few. It was Jonathan Weiss's project, undergraduate project, to sift through those and find interesting candidates and also confirm those losses by hand with synteny. So he made these beautiful synteny maps. And we noticed this one gene called PLAT1 that was lost in all of squamates, so lizards and snakes. This piqued our interest because PLAT1 in fish and PLAT3 in mammals has been recently discovered to be responsible for lens clarification, so having a clear lens in your eye. And of course, Suzanne works is a cavefish biologist, so we're interested in visual evolution. And also because squamates, lizards and snakes, have a pervasive pattern of visual gene loss across their history. So we thought, what is this PLAT gene? What does it do? So digging a little bit into this function, in order to have a clear lens, or in order to see well, you have to have a clear lens. If you don't have a clear lens, that's a cataract. And in order to have a clear lens, you have to get rid of all the cells in your lens. So you first have to burst the cells, and then you also have to burst the organelles, and that's where PLAT1 comes in. So PLAT1 attaches and finds those organelle membranes, breaks them open, and starts the process where macrophages and stuff can come in and clear that out. We also know, PLAT1 does this in fish, and PLAT3 does this in mammals, we think. There's some overlap in function. So we also know a little bit about what each domain of PLAT1 is doing. They have different functions. So there's this, wish I had a pointer, there's this transmembrane region that is responsible for finding the organelle and attaching to it. And then there's this LRAT region, which is the lecithin retinal acetyltransferase domain, which I'm not going to say again. So that's LRAT, and that does the catalysis of the membrane, so it breaks it apart. So we know that PLAT1 knockouts in mice, or PLAT1 knockouts in fish and PLAT3 knockouts in mice will cause cataracts. But also, if you just knock out the transmembrane domain, that little N piece, that will also get rid of the lens clarification function and cause cataracts. So it's extremely vital to the function. We also know there's 5 paralogs in human, and there's paralogs elsewhere in the vertebrate tree. So we don't really know what exactly PLAT1 is doing across vertebrates and outside of just zebrafish and mice, really. So we know it's related to the physiology of the eye, and we know it's got this loss at the base of squamates. So our bigger question here was, does PLAT1 degradation underlie vision loss in blind vertebrates? And to answer that question, I swooped in and used NCBI ortholog database to get all of the PLAT sequences I could from mammals, fish, and sauropsids, which include many blind mammals as well as blind fish, usually cavefish. We also sequenced 13 novel cave fish genomes. Some of them are cave fish, some of them are blind with other physiologies. For the sake of this talk, they're all just going to be blind. And of course, cave organisms are adapted to low light, so they're all low visual acuity and blind species with a range of like completely lost eyes versus they just can't see very well. And we're looking in those species, we would expect to see signals of selection, gene degradation, or true and complete loss. And just as an extremely quick reductive review, this is a reminder that relaxed selection has the same direction of signal as positive selection. We have some cool mathematical models that can tell them apart, but there's almost always overlap in those signals because those models aren't perfect. So what do we see in fish? We see in blind fish that there are some branches with signal of positive selection and this very weak signal or marginally insignificant signal of relaxed selection, which is not super interesting. However, that is not all that is going on with fish. If we look a little bit closer in fish, so tests of selection can only detect sequence differences and changes in codons, but they are totally blind to deletions. And what we found was that in these blind fish, there are 3 lineages with premature stop codons that completely knock out 2 domains that are functional, and 3 more lineages with large exon deletions in functional regions. So something is going on here with the cave fish. You don't see that in any of the other lineages, so if you have a deletion, you are a blind fish. So if we look even closer at the deletions in the middle of the exon, the cool thing about the ones with the premature stop codons is actually the sequence after the stop codon was not degraded so much that we couldn't see it anymore. It was clearly like a recent PTC. So we pulled out that sequence and we reran our selection analyses. And of course, we found what we expected to find, which is a very increased signal of relaxed selection that's centered around that transmembrane region at the end of the protein, which is, again, responsible for lens clarification function. So what is going on with PLAT in fish? We have major structural degradation that's found exclusively on blind lineages, exclusively in functional domains, and signal for relaxed selection and positive selection that's centered around that C-terminal functional region. So it's likely to have functional consequence. And then if we take a look at blind mammals, what's going on? So we get a little bit of a stronger signal in blind mammals. There's both signal of positive and relaxed selection, with the relaxed selection signal being much stronger. If we break that down a little bit to look at what's called evidence ratio of where that signal is coming from, positive selection is coming mostly from one codon in that catalytic region, and the rest is coming from the end of the protein, again, that transmembrane region. So that's likely where the relaxed selection is coming from. We also looked at PLAT3 in mammals because, remember, again, in mice, PLAT3 was thought to be the one that was doing the lens clarification in mammals. So we would expect maybe a stronger signal in mammals for PLAT3. But we did not find that. We did not find evidence for positive selection, gene loss, or relaxed selection in PLAT3. So something interesting going on in PLAT1. What is it? Strong selection for relaxed selection in blind mammals on PLAT1, and some positive selection too, likely working on the same gene in different sites. And the locality of that relaxed selection is really in that transmembrane region, which again is likely to have a functional consequence of knocking out lens clarification. So stepping back to whether gene loss is underlying convergent vision loss in these vertebrates, we have deletions that are found only in blind species among fish, and that happens across 6 independent lineages of blind fish, and only in functional domains and in nowhere else in teleos. We also see that this blind phenotype is strongly associated with convergent relaxed selection in the transmembrane domain, which is functional, and that happens across 16 lineages containing 28 species. So it's clear that PLAT1 is likely on the path to gene loss. We've got signals of relaxed selection, loss of function, and potential pseudogenization which is important for the evolution of this gene, but also because we didn't really understand what PLAT1 was doing in mammals. It's clearly still important for vision in mammals, and that has medical and veterinary translational implications for this gene as well. So just stepping back, combining these insights from gene loss, convergence, and selection can provide these large-scale insights into gene family function and targets of evolution. And with that, I will happily take questions.
Very interesting. Do all of the blind cavefish show the same degree of sort of blindness? I mean, there's some that just completely lack the organs at all, and I would imagine others with somewhat developed organs. Do you see any signal there? Because it seems like it's probably some upstream process that's leading to the lack of an eye as opposed to blindness related specifically to cataracts.
Yeah, I think that's a good question. It's all the scale of blindness is across the board. So it's a good question, though, that things that maybe still have some visual capacity have this deletion, which I actually didn't think about. I was thinking about things that like are all blind. So we have a lot of completely eyeless fish in there. But there are some lineages that just have a totally intact PLAT1. At least at the species level. We haven't looked at population level yet, but I think with most traits that are evolving via relaxed selection, there's some parallel evolution, but they're hitting all different targets in that cascade. Yeah.
So to piggyback off your question, do you think the location and size of the deletion affects the amount of vision loss in fish?
You know, actually, I don't think so. Because with proteins, as you might know, you can screw up one bit and then it just refuses to fold, if anybody's have ever expressed proteins before. So I think it's just a matter of luck. And certainly the ones that are deleting the whole domain is a pretty sure bet. But yeah, I think that that's what makes this kind of difficult to call loss of function. I mean, you can call the really obvious ones, but all it really takes is one bad stoichiometric interaction that'll destroy it. So, yeah, thank you. Any other questions? Thank you so much.
Hello everyone, I'm Yen-Wen Wang. I usually go by Danny, and I'm a postdoc researcher at Yale University under the supervision of Jeffrey Townsend. And our lab usually do cancer stuff, but we also have a proportion doing fungal research, and I'm going to share with you about the parallel evolution that we are investigating in mycoparasites. And because this is a mostly animal and plant conference, so I think it would be nice to introduce a little bit of fungi. And the reason why we study fungi is because the fungi are cool, and the fungal ecology is very diverse. You have the wood decomposer or litter decomposers that degrade wood and litters. You also have insect pathogens, for example, cordyceps, you probably already know from Last of Us Netflix series. There are also some mutualistic fungi with plants or animals. But what we are interested in this project is mycoparasite. And mycoparasite is the fungi that parasitize on another fungus. And so if you look at the arrow there, there's a little hypha that wraps around the bigger hypha. And that little hypha is a mycoparasite that try to attack the bigger hypha. And this is quite an important species or genus. It is called Trichoderma, and it has a pretty broad host range. can attack a lot of like plant pathogens, so it's very useful for biocontrol. There are some other microparasites. For example, this is lobster mushroom. It colonizes a white mushroom, and it can turn it into orange and make it very delicious, apparently. There are also snow fungus, another edible mushroom. And so these microparasites are very diverse. But even if we look into one single class of fungi, for example, here is Sordaromyces, we can see a bunch of different microparasites. And if we, for example, if we zoom in here, you can see that the yellow pies, those parts are microparasites, and it includes Trichoderma over at the bottom, and you can see that it evolves from the red pies. The red pies are the insect pathogens. So this is a niche transition from an animal pathogen to a microparasite. And there's another species up there, which is Tolypocladium. You cannot see the yellow pie because I didn't label the tip, but it also evolved from animal pathogen. So we have these two lineages that evolve independently, parallelly, from animal pathogens to microparasitism. And so, given the evolutionary history, we are wondering if we can use this information to find microparasitism-related genes. And how are we going to do that? We first assume the gene expression all across the genome follows a stochastic manner, just like your normal continuous traits. And so you can model these gene expression with different kind of stochastic processes. And here there's Brownian motion processes on the left, and that one would be just completely randomly evolving from one value to another value given variance. And then there's a Ornstein-Uhlenbeck process, which is similar to Brownian motion process, but it has a slight constraint that it will sort of move toward trait optima. And the Ornstein-Uhlenbeck process would sort of mimic selection scenario. And so that is very useful because if we look at the gene expression evolution, you will see that probably most of the time it will evolve in a Ornstein-Uhlenbeck process way. But when these organisms go into a different ecology, these genes will under a different selection pressure, and it can move the gene expression optima from one to the other. And so we can try to identify the microparasite if there are any genes that parallelly evolve from one optimum to another optimum, and that would indicate these genes are important. And so we are applying this modeling system to microparasitism. And what we did was we selected 6 different species. 2 of them are microparasites, including Trichoderma aspergilloides and Tolypocladium ophioglossaides. And we also include 4 different non-microparasites. These are just basically make a baseline of this evolution process. And so if we put it back into the tree, you will see that the Trichoderma aspergilloides and Tolypocladium ophioglossaides, they both evolved from animal pathogens independently, and they all diverged actually quite long ago, more than 100 million years ago. And having this study system, we designed our experiment to focus on spore germination. And the reason why we chose spore germination to study is because spore spore germination is the first mean of these fungi to contact to their host. And so we sequenced 4 different stages of spore germination: fresh spore, polar growth, doubling of long axis, first hypobranching. And we use these sequences to fit to the Orton-Uhlenbeck model with these possible expression level optimum changes and try to identify if there are any signals of parallel evolution. And so just a reminder, yeah, we are looking for optimal gene expression level change in trichoderma and Topoidium branches. And so what did we find? We found 27 genes that have signals of parallel evolution. If we zoom into one, we can see that the trichoderma and Topoidium has a very different expression pattern from their ancestor and the extant non-mycoparasitic species. And so this is a good first step. And later, we are planning to do some molecular work, but because 27 genes are a little bit too many, at least for our non-model system, so we are shrinking it down by categorizing them with 3 different statistics to find the more interesting genes. The first one is degree of parallelism, which is how much it evolved from the ancestral state. And then the second one is expression fold difference. And so we sequence another set of microparasite, but we grow them on a mushroom powder media that simulate a microparasitic environment. And we are interested in if there are genes that are upregulated or downregulated in this simulating environment. And lastly is correlation of expression pattern. We compare the simulated gene expression pattern, the gene expression pattern under the simulated environment, and also the original data. The reason why we did this is we want to check if the original data set actually represent microparasitism scenario. And so if we plot all the genes into this 3D plot, you can see there's a wide diversity on this 3D landscape. And we decided to just cut the cube in half to find which genes are probably more interesting. And so we can see there are the red ones are having these higher value of these 3 statistics. And these will be the first genes that we investigate. And because of the time limitation, I'm probably just going to talk about this gene. This gene is AMDHD2, and you can see that it has a parallel evolution in trichoderma and Topoidium. The gene expression pattern is quite interesting. It expressed a lot at this first stage. Oops. Yeah, it expressed a lot at the first stage, and then it quickly went down afterwards. If we look at the simulated environment, the solid line, so the most right-hand side plot, the solid line is a simulated mycoparasitism environment, and you can see that at stage 2 to 4, it's upregulated, but not stage 1, which is also quite interesting. And so because we are just using a simulated microparasitic environment, we decided to look at other literatures if there are any evidence of it's upregulated. And it turns out when we look into 5 different literatures, actually 4 different literatures, one is from our own lab, and we can always see this gene being upregulated during microparasitism. And so this is an evidence that this gene is probably associated with microparasitism as well. And what does this gene do? So this gene encodes an acetylglucosamine 6-phosphate deacetylase, and this is an enzyme that's involved in chitin degradation. And as you probably all know, that the fungal cell walls consist of chitin and other saccharides, and so it is quite important for these fungi to produce these chitin degrading enzyme to start the microparasitism, probably. And so I hope that this convince you that our method can detect microparasitic related genes. And so right now, I'm just going to skip all these slides on other genes. Okay. Right now, we are doing some network analyses, trying to find some gene regulatory networks that are parallelly evolved in the two microparasites, and hopefully we can get some hypotheses here. But we don't actually know that much about gene regulation in these non-model systems, so a lot of more work needs to be done to interpret them correctly. And so in summary, we have this tool using parallel evolution to find interesting genes in different ecologies. We can use, we found MDHD2 is probably crucial for microparasitism. There's network analyses that probably can suggest hypotheses on parallel evolution and expression regulation. And we are also doing experimental validation for confirmation. And thank you, everyone. I'll take any question.